Hi everyone, I hope you had a decent week so far. I do apologize for this lecture also being a little bit late. My dog has been sick for the last few days, so I've been trying to take care of him and also cleaning up subsequent messes. But regardless, let's move on. Today we're going to be talking again about genetics and inheritance, but more specifically about mutations and sex-linked traits. So some key terms that you need to know. A chromosome is just a tightly coiled strand of DNA within the cell nucleus that codes for what makes every one individual. Within chromosomes, you can have autosomes, which are going to be your 22 pairs of chromosomes aside from your sex chromosomes or your X and Y chromosomes. You can have autosomal traits, which are controlled by genes on one of the 22 pairs of autosomes. But you can also have sex-linked traits, which are traits that are controlled by genes located on a sex chromosome. Sex-linked traits have recessive alleles on the X chromosome, and because males only have one X chromosome, if they have the recessive allele, they will express the trait. Women tend to have a little bit more protection. When you have two X chromosomes, typically your stronger X chromosome or your one with the dominant allele will mask the recessive and you won't end up getting the disease. That is why women tend to be carriers more. And this is what people are talking about with like male pattern baldness, when they say to look at, the mother's father to see how that person might be, that son might end up being bald, rather than looking at the father or the father's father. So mutations are changes in the genetic code that creates entirely new genetic material. This is really, really important because mutation is the only driver of evolution that creates a new source of alleles. Mutations can be caused by multiple things. They can be spontaneous mutations, unplanned, unpreventable really, they just happen. Typically, these are going to be DNA copying mistakes made during cell division. But you can also have induced mutation. So these are going to be things like exposure to ionizing radiation. The best example of this is skin cancer from sunburn. It could also be exposure to chemicals called mutagens. And these are going to be things, oftentimes you can see these when you hear about a chemical being linked to cancer. That would be a mutagen, and it causes mutations that can lead to cancer. But you can also get, or a mutation can also occur due to infection by viruses. That is also an induced mutation. So here at the top of this little image, this is a point mutation where thymine, which is represented by that letter T, is being replaced by cytosine. And as a result of this change, valine is the amino acid being produced. If this change hadn't happened, then isoleucine would be produced instead. And cytosine, guanine, adenine, and thymine are base pairs. They're kind of the building blocks of all proteins, and they're called base pairs because they are paired together. So if there's a change in how these are paired, then it can have consequences. Now, sometimes point mutations can result in the production of the very same amino acid and the very same protein, so they obviously have no impact on the organism's phenotype. When this happens, they're called synonymous point mutations, and they're fairly neutral mutations. They don't really have much of an effect because they're not really changing anything. We just know that the amino acid has been changed. It's not, things aren't functioning exactly how they normally would. The example here, though, in this picture, you can see it's these two. This is actually a non-synonymous point mutation because a different amino acid is being produced and therefore a different protein is being coded for. And this can have a really dramatic effect on the organism. Now, here at the bottom is another kind of mutation. This is called a frameshift mutation. And in these cases, a nucleotide is actually being inserted into the gene and shifting down all of the remaining nucleotides by one. This is important because nucleotides are actually read in groups of 3 called codons, so shifting everything down by one actually dramatically changes the DNA sequence and how it is read, and it results in entirely different amino acids being coded for and entirely different proteins. So these can have a much more negative effect or a much more drastic effect on the individual. This can occur because genes can copy themselves and be inserted into different regions of the genome. When this happens, they're called transposable elements. But what's really important is that synonymous point mutations are relatively uneventful. They're typically not negative or positive, they're more neutral mutations. Whereas a non-synonymous point mutation is a little bit more dangerous, can have more negative effects. And these frame shift mutations are the most dangerous, are much more likely to cause significant problems. You can also have mutation due to duplication. This is what is called trisomy, which is a genetic disorder where a person has 3 copies of a chromosome instead of the typical 2. These can be sex-linked or autosomal. An example of a sex-linked trisomy is Klinefelter syndrome, where a male is born with an extra copy of the X chromosome. Anytime somebody is born intersex, they typically have some form of trisomy where their sex chromosomes are not your typical 2 base pair. So instead of being XX or XY, these individuals are going to be XXY or XXX or XYY or whatever other combination. But you can also get it on autosomal linked traits, so like trisomy 21, where you have a extra copy of the 21st chromosome, is going to result in things like Down syndrome. This is used a lot for, a good example of this because it's something that most people have seen or have had a personal relationship with at some point. It's also a fairly obvious one to see phenotypically. Now, obviously, these are considered negative mutations because they create something that is more abnormal. It's not considered standard. But just because it is considered a negative mutation does not mean that it's going to have completely disastrous effects on the individual's way of life or their quality of life. Obviously, people with Down syndrome and Klinefelter syndrome can live very happy, very fulfilling lives where they aren't constantly in pain or suffering from their disease. But they're still considered negative mutations because it is abnormal and it does cause the individuals to have extra hardships that they might not have otherwise. Now, you guys do need to watch this YouTube link that I have in the lecture. It'll explain mutations and sex-linked traits a little bit further. And you should do the chromosome activity. It'll show you how to kind of match up chromosomes and tell how studies figure out which ones are paired and which ones there are duplicates of. But it'll also give you like case studies for each round where you can see how having some type of mutation, I think most of them are duplications, but it'll show you and explain how having that duplication or mutation has affected the lives of the individual with the disease. You should also do this color blindness test. It's really just for fun. There's no way to get it right or wrong. But screenshot your results from that and submit them with your lab assignment just so that I can see that you guys are participating. Your lab 3 is due on February 19th. You also have your first quiz coming up soon. This will be due on February 23rd. But a few days before it opens, I will send out an announcement through Blackboard, so be on the lookout for that during this week. It'll go into a lot more detail about what you guys should expect from this quiz, what'll be on it, how long you have, all of that kind of stuff. Now, I have gotten quite a few emails about some confusion with the Lab 3 assignment. That's totally to be expected. Let's see. So we are going to do it together because I know that sex-linked Punnett squares can be a little bit daunting, especially the first time. All right. So if you go to this first problem on your guys' Lab 3 assignment, you're going to see a bunch of text and then a Punnett square. Make sure that you read the text because it does give you some clues on how to do the Punnett square. So for instance, it says that red-green color blindness is a recessive sex-linked genetic disorder. So we know that it is recessive and sex-linked. And that's going to help us out when we're doing our Punnett square. So because it's recessive, you're going to have to choose, just like any of your other ones, how you're going to label the trait for this disease or this disorder or, whatever. I'm going to choose G just because it's red-green color blindness, but you can choose whatever letter you want. And per usual, this one is going to be dominant, and this lowercase is going to be recessive. Because this is a recessive disorder, we really only care about this lowercase g. This is what we're going to be on the lookout for as we complete our Punnett square. So when you continue reading the problem, at the end you can see where it says that we are crossing an unafflicted male and a female who is a carrier. So that gives us a clue about the parents' genotypes. So parent 1 is going to be this unafflicted male. Because he's unafflicted, he isn't going to be a carrier, nor is he going to have the disease. And it is sex-linked, so we still need to use XY for male and XX for female. But because the man is not a carrier and he's not suffering from red-green color blindness, he's going to get this dominant allele, and it's going to be on this X chromosome. Now we know that the mother is a carrier, so we're going to have one dominant allele and one recessive. Now we'll complete our Punnett square like we normally do. We'll just fill all these out, read them like normal. Now you'll see that it also asks you to find your genotypic and phenotypic ratios. And your phenotypic your genotypic should be pretty easy because obviously all of your offspring in your Punnett square have a different genotype. Every single one of them is different. So you know that it's going to be 1 to 1 to 1 to 1. So we've got 4 total. Each one is different. Now your phenotypic ratio might feel a little bit more confusing because you're going to have unafflicted individuals that don't have the disease and they're also not carriers. You're going to have carriers, and then you're going to have those that are red, green, colorblind. So our carriers are probably going to be one of the easiest to pick out because we only have one offspring that is a carrier, and it's got the exact same genotype as our carrier mother. We're also going to look for our unafflicted offspring, and that's going to be these top two, because there is no recessive allele for the trait anywhere, so they can't end up inheriting that trait because it's not present in their genotype. That makes it pretty easy to figure out which of our individual offspring is red-green colorblind. It's going to be this male that does not have a dominant allele to mask the trait. It's only got that recessive. Now, because of this, you guys might be tempted to do your ratios as 1 to 2 to 1 for your phenotypic ratio, but we are not going to do that because we actually don't care that these genotypes are different between unafflicted and carriers, nor do we care what sex the offspring is. So we don't care that this one over here is male while these other two are female. It doesn't really matter because we're only focused on the disease. And because we're only focused on the disease, we're going to lump these three together because outwardly speaking, physically, if you were talking to any of these three people, you would not know whether they were a carrier or if they were unaffected by the disease. When we lump these together, we're going to get a P ratio of 3 to 1. We have one colorblind individual and three that are not colorblind. We don't care that if they're completely uncolorblind or if they're a carrier, it doesn't matter. We only care that they aren't colorblind. All right. That is all I have for you guys today. Obviously, if you have any other questions, let me know. When you're doing the rest of this Lab 3 assignment, especially when it's asking about harmful or beneficial mutations. We talked a little bit about it last week, but you guys, if you need to, do some outside research. But basically, it's pretty simple. If they're beneficial, it's going to increase your chances of adapting to environments or disease prevention or protection. It's going to create some kind of reproductive advantage or tolerance for food or something like that is going to benefit the individual and allow them to continue to reproduce. So it's going to be this idea of natural selection. If they're harmful, then they're going to create things like genetic disorders, cancer, developmental issues, health issues, sometimes even death, just essentially reproductive failure. You're not continuing your population one way or another. Or if you are, they're not the best adapted for their environment. When you're doing your essay question, choose from any of the genetic disorders. Whatever looks the most interesting to you is fine. If you choose lactose tolerance, be very careful. You need to be researching lactose tolerance as a mutation, not lactose intolerance. That's not a mutation. Humans are naturally lactose intolerant once they get past the weaning phase with their mothers and breast milk. So I don't really care about knowing more about lactose intolerance. I want you guys to be looking into lactose tolerance as a mutation that occurred that allows certain groups of people to continue to drink milk past weaning age without any kind of issue. So just keep that in mind if that's the one that you choose. I don't want you guys to be losing points off. I know that it's an easy mistake to make. That is all I have for you guys today. Let me know if you have any more questions. I do think we're supposed to get some more cold weather soon, so bundle up if you're going to be outside, drive safe. And I will see you guys for next week's lecture.